class of cancer-described traits that govern the transformation of normal cells to cancer cells. One of these hallmarks is enabling replicative immortality, of which telomeres play an important role. So what are telomeres, what do they do, and how are they involved in replicative immortality? First off, there is the control of telomere length. Within each cell, there are 23 pairs of chromosomes which carry our genetic material. At the ends of these chromosomes are regions called telomeres, which are TTA-GGG repeats. They protect the genetic material from degradation. As with each cell division, the chromosome shortens in length, and instead of losing the important genetic material, the telomeres are lost. And eventually the telomeres become so short that the cell will stop dividing and enter senescence, which is a permanent state where cells stop proliferating but do not start dying. This usually occurs after 30 to 50 cell divisions. Telomerase is a ribonucleoprotein which uses its own RNA template to add TTA-GGG repeats. Therefore, it maintains or extends telomeres. And whilst normal cells have telomerase, cancer cells have higher levels. Another mechanism to obtaining or extending telomere length is alternative lengthening of telomeres, or ALT. And this is only used by cancer cells. Although it's agreed that ALT requires a DNA recombination step, the mechanism of lengthening is uncertain. Two have been proposed. The first is unequal telomeric sister chromatid exchange, or TSCE, and the second is homologous recombination, and these are not necessarily mutually exclusive. In unequal TSCE, after DNA replication, there's an unequal exchange of telomeric DNA between sister chromatids, resulting in a lengthened telomere and a shortened telomere, provided there is a mechanism for segregating the two so that all the lengthened telomeres are in one daughter cell and all the shortened telomeres are in another. There could be unlimited proliferation of the cell population. In homologous recombination, using an existing telomeric sequence as a copy template, there is a stand, invasion, and synthesis of new telomeric DNA. Therefore, is a net gain in telomeric length and an increased capacity for cell proliferation. So normally telomeres are deleted. This deletion acts as a biological clock for limiting cellular lifespan. However, cancer cells have high levels of telomerase and/or ALT. This aids in the transformation of normal cells to cancer cells, as it enables the cell to achieve replicative immortality.